Okay, so here's an example of that. All right, so what they've done, once again, this is another example. We'll look at this example and then we'll kind of do a problem. All right, so let's go ahead and explain our way through here. What we've got is, here is our template DNA in blue. Can everybody see that in back, or is that too small? Looks like, okay, I'm getting thumbs up. So we've got this template. This is our primer. So the primer goes back here some, the DNA goes back here some. And so for our this reaction right here has DATP plus dideoxy ATP, right? So it's going to stop at every A. This one's going to stop at every C. This one's going to stop at every G. This is going to stop at every T. And when I say stop, I mean it'll make a product for every one of those if you've got your concentrations correct. It won't actually stop at every A, because if it's the first A it stops at, it's done, right, for a given product. But so for this reaction, if we look up here, we've got, if we're going across from it, we've got 1T here where it'll stop and 1T here where it'll stop. And so we have two products down here that end in A. Okay, over here we've got a C, so how many products should we make based on the template? 3. We're going to stop here and here and here. And actually we would run to the end, but they're not really showing that at all, right? You just keep going until it ends as well. So, but all the other products. But here they're just showing for this, I guess. This shouldn't be here, actually. That's confusing, because there's a whole bunch more it would make beyond that, right? There's got to be some more A's, G's, and C's, whatever there. But for this one, 2 products, right? That's going to stop here and there, okay, for what we see. And for this one, we're going to stop here, no, T across from A. We're going to stop there, there, and there. That makes sense to make those products? And then we would run it out on a gel. Okay, and looking at this gel, you can look at the gel here, but you can look at the products as well. What's our shortest product looking up here? It's the GGC. There it is. And our second shortest product, this one, there it is. Our third shortest product, this one, there it is. So we would just read it up. Does that make sense? Okay, let's try one. So let's say a DNA sequence DNA template has the sequence let's go 5 prime A G C T T A G G C T A G C G C G A T C. I'm typing, I win. Okay, so it is sequenced using the following primer. Okay, let's say our primer goes 5 prime CGC A. Okay. Draw all products for a reaction that contains DATP, DCTP, DTTP, DGTP, and DD, so dideoxy, this is the one that stops it. Let's go ATP. Go ahead and take a couple minutes and try that. And call me over if you have a question. And I have not, remember, things have to be anti-parallel. And I haven't drawn it to be easy for you, unless you like read Hebrew or something like that.
Can you make it bigger?
Yes. I'm going to have to custom this. Let's try 60. Is that good enough? Yeah. Okay. That's what goes in the reaction. So those are the nucleotides that we have, right? First thing, hybridize your primer. How many people are confused on contains the DC? It's okay to be confused. Okay. And I'm just going to go talk to Sophia. If you're confused as well, you just missed out. Anybody else want to raise your hand? Okay. So, this. If you're going to add a base, you have to extend it. You have to stick in nucleotides, right? So the DNA polymerase has to fluorize by just choosing dideoxy-GA. It will stop at the first T if that was the right thing after the primer hold. Because we don't get anything. And then starting back such that not even the mix and there's some natural mix that makes it so that it doesn't put all I was just going to start with so you can get it in for a long way so you can see how it's supposed to. Okay. Does that make sense? I just yes, try to help each other. But I'm not going to give you a couple more minutes. Maybe. Okay. So let me go do. Okay, so I'm gonna go ahead and do this one. I am going to scoot this sequence down so I have some space on top. Okay, and so can you guys see this on the bottom? Yeah. Is it too low for Hugo? All right, so actually let me go up just a little bit. There we go. All right, so I need to nail this primer somewhere. It has to go anti-parallel. I think I'd rather rewrite this one than rewrite that. So I'm going to go, but it's going to go anti-parallel. It's going to match up somewhere here, complementary to this. I know it's right here because I designed it, but you need to be able to kind of look at it and figure that out, right? You need to be able to look at these sorts of things. So it goes C, G, C, A, 5 prime to 3 prime. You guys see that? Okay, so that's where my primer is going to sit down. I'm going to write my products up above it. So I'm going to have a 5 prime C, G, C, A. Now remember, we have dideoxy. We have all the 4 of the nucleotides we need, plus we've got dideoxy A in here. So it's going to stop wherever I add an A, for not every A, but just occasionally it will stop where I have an A. So I'm going to go, if I look down here, I've got a T, right? I've got a C, a T, a G, and there's a T, so I could stop at this A. Okay? So there's one product. So what were those other deoxy things about? When my DNA polymerase came in here and came to the end of the polymer here, it went out and it felt around in solution. It tried a bunch of different nucleotides. It grabbed a G, it didn't work. It grabbed a C, it didn't work. It grabbed a T, it worked. It added a T. Does that make sense? Okay. It grabs around solution, it finds a C, it puts it in. It grabs around solution, finds a C, it puts it in. It grabs around solution, finds a T, it puts it in. Okay, so we can keep adding these things and keep going because we have those reagents in solution. Gets to here, it grabs around solution, it grabs a dideoxy-A instead of a deoxy-A. It puts it in. Can't extend anymore. But there, yeah, there's going to be millions of products being made, billions of products, probably more than that even. So this thing, here's my primer. We're going to go ahead and add the stuff we added before. Okay, in this case, it reaches around in solution, it grabs an A, it turns out it's a deoxy-A. It can keep going. Okay, so then it adds the next A. This time it randomly grabs a dideoxy-A. It's done. Okay, yes?
We didn't have all four deoxys. Does it, like, say we didn't have, like, the G one, does it just immediately stop at that?
That's a fantastic question. Did everybody hear that? If you don't have all the deoxys, you get to where if it needs a G to continue and there's no G's, it's done. It doesn't add anything more. Okay, and so one way I might test this is I might drop one of those deoxys and I want you to look at it and say, okay, well, stop there, I'm all done. Okay, excellent question. And then for this one, if we were looking at it, so the primer isn't really a product, although the primer would be in solution, but we wouldn't probably see it because we hadn't incorporated anything. But then we would go ahead and our final product would go CgCa TCCTG. Here it reaches around in solution, it finds a deoxy-A, so it puts it in. It reaches around in solution again, it finds a deoxy-A, so it puts it in. So it can keep going, and now we're going to go GCT, and for this sequence, now we're done because there's no more template, right? Does that make sense? Why I kept going till the end?
So there's 3 products?
So there are 3 products there. The 2 that stop at the A's in the sequence, when that's added, and then one that just goes to full length.
And the shortest one we have would be at the bottom of the gel?
Yes, so if we ran a gel, we'd have three bands. I could draw it, but it doesn't really matter because I don't have other bands to compare to, right? I mean, if I was to draw it, it would look like this. I'd have one here, I'd have one here next to it, and then what is it, 3 more up, and maybe have one here. These gels aren't really linear. They get more, you've seen this before. You've seen this in our gels that we ran. They get more compressed as you go up. It's a logarithmic scale. Okay, questions? Let's try another one. I'm going to hand out a worksheet, and I'm going to post the answer. And you are welcome to work this worksheet. I've already printed them, so that I'm not going to lose them before next year. So go ahead and grab a worksheet. I will post an answer key for that, and that might be something you want to practice. But let's do one more of these. Draw all products for a reaction that contains deoxy ATP, deoxy GTP, deoxy CTP, deoxy GTP, and let's see, dideoxy CTP. Okay, we've got same primer.
Isn't one of them repeated?
Yes. So you could answer, that's not what I intended. So I'll change it, but that could be your problem, right? And so if that was the case, where would this thing stop? The first time, the way I wrote it, it would stop the first time there was an A on the template or it required a T to keep going. But I'll fix it. Because that wasn't what I meant.
It's just an extra.
And call me over if you have questions. Lorenzo, I've got the primers working okay.
Okay.
On the worksheet, Lorenzo pointed out there's a mismatch in the middle of it, of the DNA sequence. There's a C across from a T. We'll just call it DNA damage, but it doesn't really affect the exercise. Okay, so let's talk about what the products would be. So once again, we're going to hybridize that primer down here. And so I'm just going to put it on here so I know where I'm going with this. So I've got a C, a G, a C, and an A, right? And then I'm going to polymerize from that. Our dideoxy here is a dideoxy CTP. So we're going to go out 2 and stop, right? So I'm going to go C, G, C, A. I'm going to definitely, since I'm writing it this way, I'm going to definitely make sure to mark what my 5' and 3' end are. And then it's going to go T and then a C and stop, right? That'll be one of mine. Then I'm going to have the same thing again, C, A, T, C, and it looks like C and stop, so one longer. And it looks like then we're going to have another one where we go, we're going to have 4 products. C, G, C, A, T, C, C, and then I'm looking at the top here, T, G, T, T. I've got a C to put in, oh, it's across from the G's, a G, and then I've got a C and I stop, right? And then finally, I've got something that goes the full length of the sequence. So C, G, C, A, T, C, C, T, G, T, T, G, C, T, and then we're done because we fall off the template essentially. So I was more focused on making sure I got the right answers there than pointing out where I was doing it based on time. We've got a minute. Does anybody have questions on my 4 products?
You wrote TT when it's AA.
Both, like all the times?
Just the last 2. It's B,
The last-
A, G.
This should be A.
Yeah.
Thank you. Any other questions? That was a comment. Thank you, though. Okay. All right. Well, you guys have a good weekend. I will post